Alright guys, so today I'll be going over how to do the bioinformatics stuff. The first part of it is finding your amplicon. And the purpose of finding the amplicon is to show the region that we're scanning for mutations. So today I'll be doing it for ebony. So this gene right here. So let me show you guys how to do it. So let's just say your mutation is ebony. The first thing you want to do is go to Flybase. And go to the ebony gene. Right there. And the next thing you want to do is go to get decorated FASTA. And here's your sequence. So copy this sequence. And then go to Microsoft Word, paste the sequence, and the next thing you want to do is you want to find and replace. So the shortcut for that on Windows is Control H. But yeah, just get to find and replace, and what you want to do is you want to find all of your caret P, and you want to replace it with blank. So replace all. And what that does is it gets rid of the enters at the end. So if you go back, each line is separated by an enter, and what that does is it gets rid of all of that. So now you have your sequence of your ebony gene. And also things to keep in mind, so the CDS or the coding region are in pink and the untranslated regions are in blue. So that's really useful when you start highlighting your amplicon. So all this is protein coding and all this is non-protein coding. So the next thing you want to do is you want to go back to your primary list and you want to start looking for these primers. So here's ebony, group 26, okay. We're going to look for this primer sequence within our sequence. Control F, Control V, and then we couldn't find the primer sequence in here, which is alright. So what you want to do next is you want to go get the reverse sequence. And you want to Control F, Control V, find next, and there it is. So this is where our primer sequence is. So you want to go home, highlights. So this is where this is one of the ends of our amplicon. And what we want to do is we want to find the other end. So as we know, we can't actually find the sequence in our ebony gene. So what we're going to do is do reverse complement. Here it is, bioinformatics.org. So highlight it all, put your sequence in here, submit it, and this is your reverse complemented sequence. And then you're going to copy this, and you're going to find it in here. And there it is. So go home, highlight. So now you know that from here all the way to here is your amplicon, or at least for the purposes of our paper, is the region that we're going to be scanning for mutations. So what you can do is you can actually copy this out, make a new page, paste it, and then things that you can do is you can mention that the region that you're scanning has 2,380 nucleotides. Okay, so the second thing we'll go over is the Finch TV stuff. So first thing you want to do is you want to actually, I'll show you guys where it is. I'm going to go to Blackboard. You want to go to course info. Then you want to go to Bioinformatics documents and Finch TV downloads. Here's Finch DNA sequences. And then you want to go to your the class that you're in, and then so this is my class group 26, and then this will download a zip file with all the Finch TV files. What you want to do is you want to copy those out into a folder or something so you can access them. So here they are. This is what's inside of this folder. So I'll do a few of the examples with you guys, or to show you how to do this. So this is probably the most confusing part. So these files, they contain the sequencing data for these mutants. So these are black mutants, and these are for ebony mutants, and these are using the black forward primer, the black reverse primer. This is black with ebony forward primer, black with ebony reverse primer, and then it's the same here. So this is ebony with the ebony primers, and this is ebony with black primers. So what you would expect is that the ebony mutants, you should see a mutation with ebony primers, but ebony with black primers, so you're looking in the black gene, those should be wild type, or you shouldn't catch any mutations. And then so for group 26, ebony is the mutant. So ebony mutants with ebony primers, if you see mutations in here, those are what's contributing to the ebony mutation. Now your complement fly is the black fly, and black with ebony primers. Of course, you would expect the ebony gene and the black flies to be wild type, but black flies with black primers, you would actually expect to see a mutant. And what this is showing is that this is to confirm that your black flies actually do have a black mutation in there, not an ebony mutation. Because when you do your complementation cross, how do you know the flies that we're actually working with are black, right? They actually have a mutation in black. They could actually have a mutation in ebony, per se. So this is kind of like a control experiment to show that your flies are indeed mutant for black and not mutant for ebony. And here is to show, or actually to help you find the specific mutations that are causing your ebony fly. So let's start with ebony. So first thing you want to do is you want to open up Finch TV. So here it is, Finch TV. And you want to drag your file. So this is using the reverse primer, and this is using the forward primer, and this is using the reverse primer. So if you remember from here, I think this was actually the forward primer. So this is where I'll start scanning for mutations with the forward file, and then this is where it's going to start scanning from the reverse file. So we'll come back to this, but let's do forward with ebony. Okay, so this is the chromatogram. What you want to do first is use the warp text so you can see more on one screen. And this sequence actually looks pretty good. So this is what bad looks like. So if you see a bunch of messy random peaks like this, that's bad. Do not use this. This is junk. This is okay, but this is not great either. What you really want are these nice clean peaks like this. So for example, I would start maybe from here, and I would go all the way down to, let's say, maybe here. So Control C to copy all of this, all the nucleotides here. And then what you want to do next is you want to go to NCBI Blast. And there's two ways to do this. Let me show you one way first. So first you want to align two or more sequences. On the top you want to paste what you got from Finch TV. So copy this and paste it into here. And then you're going to align it against what your amplicon, really. So let's go to here. So this is what you're going to align it against. And what's cool about doing it this way is you can actually use the dot plot function. So if your mutation is, say, caused by a transposon, caused by a large deletion, you'll actually be able to see it. So here it is. and blast. Okay, so we have 100% cover. So if you look at the dot plot for that, it's showing a straight line, right? And which means nucleotides from here are lining up with nucleotides from here. So the first base pair here is lining up against the first base pair here, and then it's a full 100% coverage. So what it's showing, so as you can see here, your max, it's the max score. You have 100% cover. So what this is showing is the sequence that we just blasted is matching up directly to our wild type sequence. So this is wild type sequence, which we got from Flybase, and this is what we've sequenced. So there's no differences. So what do you do now then? Well, the next thing you want to do is, okay, so if we scan, this is the forward primer. So if you scan from here, there's nothing mutated about this bottom region right here, right? But what about this top region? Is there anything wrong with the top region? So we're going to do the same thing, but we're going to be using the reverse primer sequence. So let's see. There it is. So drag the reverse sequence on here, and you can see it's the reverse sequence, ebony with the ebony primers from the reverse sequence. Same thing, this is junk. As you can tell, the peaks are all crazy looking. Generally, the beginning you'll have a bunch of junk too. So start where it starts looking good. So maybe right here. And yeah, at the bottom this starts looking pretty bad. So maybe we'll start, or we'll stop. We'll stop here. There's kind of a double peak down here, so you don't want to use this. Let's copy this. Let's go to NCBI Blast again. Just hit back. And actually this is again our wild type sequence, that or our amplicon, so we can kind of just leave this there. So select all of this and then paste in the reverse primer sequence, or the sequencing data from the reverse primer, and then you want to blast that again. Okay, so here we have a 98% cover, which means 98% of our nucleotides has a match, and you can check the alignment. So now you notice that there's two parts, right? There's a top part and there's a bottom part. There's a couple of mismatches right here. But the interesting thing is, when you look at the dot plot, notice how there's a break here, right? There's a break of a, this is 800, this is 600, so this is maybe, this is 200, so this is about a 400 nucleotide break, right? Right here. So that means you have something huge that has inserted between the sequence that you've just sequenced, right? So to explain this, this is the reverse primer, so you're scanning from this end, and then suddenly there's a huge section that's empty, that's not aligning, and then you start seeing your sequence back again. And that's what the dot plot is showing, right? There's a matches all the way up to here, and then if you notice that there's a little blank in that region right here, this is actually the mismatch that you see right here. Notice how there's a little mismatch here, so that's what you see in the dot plot. And then there's a big gap, which is stuff that is not aligning, and then it's starting to align again. So this means that there's a big insertion right here. So what does that mean? Well, if you go to Flybase and oops, let's see. So let's see if I can find the sequence. All right. Well, the mutant, the allele of the mutant is ebony1. Ebony, and then you want, and if you want to go to the specific allele, use the square brackets and go to ebony1. and here it is. And then notice that the ebony 1 is actually caused by a 412 transposon, right? And here it is, it's inserting within ebony. So that that gap that you see in our dot plots, let me go to here, is likely to be caused by an insertion of some sequence, and this sequence could very likely be your 412 transposon. And we'll actually come back to this, and I can show you guys how to even find the sequence of the transposon that has jumped in your gene. So here is how to do, for example, ebony, where you have a transposon. Okay, so the third thing we're going to look at is look at the complementation sequence. So if your mutation is actually like caused by an SMP or small insertions or deletions, this will probably be more useful for you. So let's go to here. So what we can do is look at our black mutants with our black primers. And this is kind of a little trick that I showed you guys earlier. But if you look at our allele, this is black 1. So what we can do is do black 1. And you can see that black 1 is caused by a complex substitution and a point mutation. So here the comment is 4 bases that have been replaced by an 8 base pair insertion, causing a frame shift, and then also a point mutation. Right? So let's see if we can find any point mutations within that sequence that we're looking at. So we can do the same thing. Black. Let's see. Black. Get decorated fast A. Copy the bases. And for this one, I guess we'll just put it we'll label this ebony. There it is. What we can do is highlight this region and then do the same thing. Find and replace. So caret P. Replace it with blank. Replace all. Okay. And there it is. This is your black gene. And what we're looking for are point mutations in this gene. But before we do that, let's find our amplicon. Let's see. So complete mutant primer list. Okay, here's black forward. Copy. Go to Word. Oops. That is too long to be a primer. Let's try this again. There we go. So this is our forward primer. And then, which means that our reverse primer is going to have to be reverse complemented. So here's the reverse complemented reverse primer. Whoops. There it is. Let's highlight that. Okay. So this is your amplicon, or the region you're scanning for mutations in the black gene. So notice that there's a protein coding region at the beginning and actually at the end too, and in the middle, and there's also some introns. Copy, paste, do some quick labeling. This is ebony. Okay. So same process. NCBI Blast. Actually, before we do this, we should grab the sequence from here. So this is from the black mutant with black forward primer. Again, the beginning is generally junk and the end is generally junk. So let's grab the good sequences. So let's start from here. Yeah, let's stop here. This long stretch of A's is concerning. Yeah. Let's see if we can find anything first, and then later on, if we have to, we can start looking through this region. So, NCBI Blast, let's go back. Replace the top query with your mutant sequence, or your sequencing data from the forward primer. And then here, you want to replace this with the black wild type sequence. So that's this. And let's see what we get. So we have 100% cover, 98% identity, which is good, which means we do have a couple of mismatches. And actually, now's a good opportunity for me to explain what the mismatches are. So the top is your query, and that's the first, if you remember our inputs, this is the first box. This is the sequence from our sequencing data from our mutant fly. So the mutants will be on the top, and the bottom is our subject, right? And the subject, which is the bottom sequence the bottom box of text that we've inserted, is from here, which is the wild type sequence. So what this means is the bottom is the wild type, whereas the top is the mutant. So here you can already tell there's a mismatch, which means, look, in our mutant sequence, we have a missing nucleotide, which is there in the wild type. So there's possibly a one nucleotide deletion here. Then here we might have insertions, right? So in the wild type, we don't have a T, we don't have these two C's, we don't have an A, but the mutant has the sequence. So the wild type does not have it, whereas the mutant does. And there's a couple of other ones that could be interesting. So here's another one. So this is just a SMP, a mismatch. So in the mutant you have an A, but in the wild type you have a C. So what we want to do with these sequences is we want to validate these sequences in the chromatogram and make sure that these are actual good reads, this, and these SMPs, and not just an artifact of the sequencing. So when you do the sequencing, there's a possibility that the sequencing was just not clean and you get missing nucleotides, or the peaks are overlapping and you're reading the wrong peak. So it could be just bad sequencing. So what we're going to do now is we're going to go and highlight this sequence, copy. What we're going to do is find sequence, paste, enter. Try that again. And there it is. So notice how this AACGA right here corresponds to this missing nucleotide, right? There should be an A, but there's no A being picked up right here. So notice how there's a blob in this region, and there's also these bars, okay? So how to interpret this is, the higher the bar, the more confident we are in that nucleotide, and the lower the bar, for example, the single A, we're not very confident in this A, right? And notice this dotted horizontal line. So general rule is, if the bar is above this dotted horizontal line, that means we are pretty confident, where when you have things where the bars are on the line or underneath the line, we're not really confident that that is a correct read. This could possibly be an artifact of the sequencing, just a bad sequence, right? So notice how there's this like gap between these two A's. So there's possibly like another A here. So let's go back. Yeah, so it's AAA, but we're missing an A. So these three A's right here, there could actually be three A's here, but the peaks were not properly sequenced, so they look like one blob. And Finch TV is only picking up as two A's. So we can't really count this as a, this is, I don't think this is a deletion. I think this is probably just bad sequencing. And the ways that you would, explain this is that, one, that there's this really large peak that doesn't look right, and two, there's a large gap here, and then three, the bars in this region are low, which means the sequencing confidence in this region is low. So that's probably not a mutation, that's probably just an artifact of the sequencing. So let's try it for the next couple ones. So let's copy this. Let's paste that in here. So that's this. Okay, so this is pretty interesting. So if you look here, all these bars are pretty tall. They're all above the dotted line, right? And so we're pretty confident that this region is, these are good reads. But as you notice, that there's sequences in here that are not in the wild type. So these are really possible insertions. So right here, these are possible insertions that the wild type does not have, which could obviously cause a frame shift. So to present this in your paper, what you would do is you would copy this region, and you would also copy, let's go back to here, you would also copy this region and show that you're confident in this region. And let me do that real quick for you guys. So the shortcut in Windows is Windows Shift S, and what that's going to allow me to do is screenshot an area. So let's do something like this. And then with this, what you're going to do is you're going to clean this up. So that has been copied now. Let me open up PowerPoint. Okay. Let me make a new slide or two. So what I'm going to do now is I'm going to change the aspect ratio to 16:9. You don't really have to do this. Okay. So now what we're going to do is we're going to paste that. So there it is. So there's your sequence with the mismatches. Remember, the top is your mutant, the bottom is your wild type. So there's possible insertions. And next thing you want to do is you want to find that sequence in here. So actually, let's go back to NCBI. Okay, so the highlighting is gone, but what we can do is do this. So put that there. Nope, let's close this. Yep, there it is. Snap it. Okay, so we can just find that sequence. GTGTGG TACC there. So that's the sequence right here. So what you're going to do is you're going to do the same thing. You're going to crop or clip this picture. So against Windows Shift S. Actually, let me cancel out of this real quick. So make sure you don't have this all covered in blue. And then Windows Shift S. Oh, cancel. There it is. So you can always crop out a little more, and then you can delete it later. So here it is. It's been saved. I'm going to paste it. Now let's full screen this. So there you go. So your figure would be something like this. But what you're going to do is you're going to match the nucleotides. Actually, so this would match against the top. So GTG, TGGA. This is a little big. So the two C's. There we go. So what you can do now is format this, you can crop it. And this is what you would show for your paper. Now notice this G is cropped kind of bad, so what you can actually do is just clip off the so just hit crop, just cut off the G. Crop. And then notice how there's a little G right here. Just go to size and position, right click size and position, and then there should be a way to crop it out a little more. So from the left side, just do like 0.7. So for your paper, make sure everything is neat and very neat looking. And what you can do is you can actually even highlight this region like this. Do maybe shape fill, no fill, and then shape outline. Do something that's recognizable, something bright. Orange is good. Yeah, orange is pretty good. So something like that to highlight this, and then you would copy, you would paste, and then maybe let's highlight the peaks. Maybe that's better. So there you go. So you would present something like this for your final figure. So let's do this a few more times for the SMPs and see what we get. So the next mismatch that we have is this. I noticed I'm just highlighting the region right before the mismatch. Just to keep things consistent. As long as you can find where it is, that's all that matters. So copy, paste, hit enter. There it is. So it's this A. This A is an SMP. So notice how there's a really large bar on this A. So that means we're very confident that this is an A in that position, but it's actually mismatching with the wild type, which is a C. So you can also include that. So let's just do that a few more times. Let's see. So that would be here. So Windows Shift S. And if you're on a Mac, there should be a way to do this also. PowerPoint. Alright, and then we can come back to this later, GCTA. So, actually, let's do it this way. There we go. Oops, couldn't find it. GCCCTAA, and then TCC. Let's try this again. There we go. So again, highlight this region using Windows Shift S. So make sure it's not highlighted. Windows Shift S. Crop that out. And actually, I just realized you only need the top part. So just paste that into here. Control V. And then later on you would edit this to look good. I just realized the bottom part is actually not even part of it. So let's do format, crop this out. Make sure everything looks neat. So this is what you want. In fact, this line at the top doesn't look good. So yeah, so for your final paper, make sure everything looks neat. That's the most important thing. Yeah, that looks pretty good. There you go. So make sure it looks something like this for your final paper. And make sure everything's consistent, right? So it'd be on the top like this. And C, T. So there's your mismatch. So the shortcut for cropping is actually Alt J P C. So we're going to go into crop mode, crop this out, and here we're going to crop this out. And let's crop out the G. That's pretty good. I like that. And here, Alt J P C to go into crop mode again. Cut out the G. Then size and position to do the final cropping or cleaning up the image. So from the left side, let's do 07, and then from the right side, let's do let's just do 107. No, actually, that's too much. Sorry. 101. So the right side needs a little more. There you go. Now you can't see that there anymore. And there you go. This would be what you would present in your final paper. And then I'll do this for the rest of them with you guys. So let's go back to NCBI. So this is the one that we didn't use because it's probably a bad read. This was a good read, and this is probably an actual insertion. This is probably an actual SMP. Let's try this one out. So again, mismatch, there's a blank right there. So we'll copy this region. There we go. And whoops. And that A is also a pretty strong A. So I would actually keep this peak also. This is most likely, well, it's a good read. So this is a confident read. The bar for the A is high. So you would do the same thing for this one. And then let's try this one. And it would be the C, I think, right? So let's go back. Yep, it's the C, which is a mismatch. And whoops. Okay, so as you can tell, the C, we have a really good bar right here, a strong peak, really confident C. So we would also include this in our final paper. So those are all possible mismatches that could actually be giving your phenotype. Now, things to consider. Some of these might be in non-coding regions. For example, this is I think this probably is in a coding region, but what you can do is copy some nucleotides here, go back to your document. So this was actually in black. Let's see if we find it. V. So notice how that insertion sequence is in a coding region, right? So this could possibly contribute to disrupting your protein. How about this one? So this might actually be in a non-coding region. Let's check it out. So this is probably here. This is also in a coding region, an SMP in a coding region. As we discussed before, it could be a silent mutation, or it can actually cause an amino acid change. So let's try this one. Yeah, and this one's in a non-coding region. Now, this is kind of homework for you guys, but if you have a mutation in a non-coding region, like an intron, could that still affect whether or not you have a mutation or not? So do mutations in non-coding regions, like this, can that give you your phenotype? So that's some homework for you guys to do. Alright, so the last thing I'm going to discuss is how to make your paper better, or how to make these figures better. So let me jump back into here. So notice how I drew boxes around the possible mismatches or the mismatches or the possible mutations. So make sure you do that for all your figures. So what we can do is probably just copy this one right here. Let's see. And this one corresponds to the A, so copy and paste, show the peak. So just make sure everything's consistent. Another thing you want to do is when you present this, you can either call it the query. That's a bit big, so let me make that a little smaller. Copy and paste. Subject. So that's one option that you have. Maybe we can do a write align. So for my students specifically, just make sure everything looks neat, because that's one of the things that I'll be grading y'all on. So what you can do, for example, is you can do this. You can arrange them, align them to the right. Actually, let's do this. Let's send it on the right. So now, whoops. Now these two are aligned on the right side, right? And this just makes it look that much neater. And this probably can come down a little bit. There you go. You can do something like this. You can underline it if you'd like. Oops. There we go. And then you can call it the query and the subject. And then just go back here. And what else you can do is call it a mutant and so you can do something like that. So just make sure everything is labeled for your final paper, because that's really important. Make sure everything looks neat. Okay, just kidding. I just realized that I did not cover the protein blast. So what I'm going to do is I'm actually going to include the footage for the protein blast in this video. So all of the bioinformatics stuff is all in one video. So the following slides will be cropped from that. Alright, so the last thing we'll do today is a protein blast. So the first thing you want to do is go to Flybase, and you want to jump to your gene. So the gene that I'll be doing today is armadillo. And go to the page with your gene, so arm for armadillo. So the next thing you want to do is go to gene model and products on the right side. And give it a second to load. so this might be a little slow. And now we're under the section for gene model and products. And the next thing we want to do is go to the section for polypeptide data. And when we go to this tab, we want to go to GenBank and pick one of the isoforms. Doesn't matter which one. And you'll be linked to the NCBI page for armadillo. And here's isoform A in Drosophila. And the next thing you want to do is do a run blast. And then you want to go down and you want to blast your armadillo gene into Homo sapiens. So the organism, do Homo sapiens. Give it a sec to do the blast. Alright, so here are the results for our blast of the Drosophila armadillo protein. And here are the different results. So as you remember from earlier, the lower the e-value, the better. So here our e-values are all 0, or they were pretty much round down to 0 pretty much. And the next thing you want to look at is your query cover. And your percent identity. So here it's showing that your percent identity, which is what matches up to, for example, here the beta-1 beta-catenin isoform 2 of Homo sapiens is a 67%. So we'll use this as an example. So we'll go to beta-catenin 1 isoform 2, and this is what the results will look like. So the top is your query, and the query is what you have inputted. So that is your Drosophila armadillo protein. And the subject at the bottom is what you're blasting against, or what you're aligning against. So that would be your catina beta 1 isoform 2 in Homo sapiens. And what's in the middle is actually the results of the alignment. So as you can see here, these are what we call gaps when there's nothing there. And 8% of this are gaps, and that just means there are no matches at all. They're completely different. The pluses are what we call positives. As you can see here, and what positives are is that the your protein on the top and the protein on the bottom are similar. So if you remember that figure from earlier with the hydrophobic and the small lipophilic, those amino acids, the positives are the amino acids that are similar but not the same. And then when you have an amino acid in the middle, for example the W and the QQ or the SYL, that means you have an exact match. So for your final paper, you need to make sure you copy or have this as a figure in your paper. Some more things to go over. These dashes here mean that it's not in your query, but it is in your subject. And this means that your it's not seen in the armadillo protein, but these three amino acids are actually found in your human catenin beta 1 isoform. And let's see if we can find the opposite. So the opposite of that would be something like this, where you have a segment in your armadillo protein, but it's not found in your human beta-catenin.